Hi everybody, thanks for having me here today. Today I just want to tell you about some work we've been doing in the epigenetics laboratory, actually for some years now, on how having an overweight diabetic mother or father can have lifelong health consequences. So in the epigenetics lab, we're really interested in the way that environment can modulate the way that our genes behave. And of course, one of the environments that has really pervasive effects on our phenotype is the environment that we're exposed to in utero. And it's been known for some years now that stress during gestation, particularly nutritional stress, can have lifelong health consequences. And prominent among these consequences are metabolic phenotypes. So just as an example, I've shown you a graph here which shows the log odds ratio for developing type 2 diabetes is higher if one is a low birth weight baby. Diabetes in adulthood, of course, is higher if one's a low birth weight baby, evidencing being exposed to undernutrition in utero. And at the other end of the spectrum, if one is a really high birth weight baby, one also has a higher risk of developing type 2 diabetes in adulthood. And so I guess our overarching hypothesis is that the intrauterine environment somehow modulates the epigenetic changes in our genome to mediate this increased risk of disease. So I guess one of the environmental stresses in utero that babies in the Western world are most likely to be exposed to is overnutrition. And this graph just shows the rates of overweight and obesity in lots of different countries. Oh dear, that wasn't what I meant to press. Oh no, you've seen all my secrets. Okay. Over the past few decades and into the future, and it comes as no surprise to see that the trend is going up. And what this means is that an increasing number of Australian women and women in the Western world are entering pregnancy overweight or obese. And I think that the actual figure is something really obscene, like in America, 2/3 of women of childbearing age are overweight at least. And so what this means is that an increasing number of babies are being exposed to maternal obesity or type 2 diabetes while they're in the womb and while they're being breastfed. And so investigating the consequences of this in human populations is sort of fraught with difficulty because of the confounding factors of genetics and especially environment. So we use a mouse model which I accidentally just showed you, called AVY, or agouti viable yellow mice. And we've been using AVY mice for some time. I'm sure some of you have seen us speak about it as a model of epigenetic variation. Because the mice carry this allele called, oh no, I've done it again. Oh dear, keep pressing the wrong button. Okay, they carry this allele called AVY, and it's epigenetically regulated and it has variable expression. So in mice in which the AVY allele is active, the mice are yellow, and in mice where the AVY allele is silent, the coat colour gene agouti is expressed in its wild type pattern and the mice are agouti. But for the purposes of this study, what we were interested in is another phenotype that the yellow mice display. They get fat, and that's because of the off-target effects in the brain of this AVY gene product, agouti. It acts in the brain to affect hyperphagia and an increased lipogenesis. So essentially, these mice get fat because they eat too much, which I think probably a lot of us humans can relate to. So this obese diabetic yellow mouse has a genetically identical twin sister over here who is lean and healthy. And so we've exploited this to look at what the differences are from being born of an obese diabetic mother or a lean, healthy mother. So we bred yellow AVY mice or agouti AVY mice to congenic males. These males carry a non-agouti allele, which is a null allele, and the reason for that is so that 50% of our offspring would also carry a non-agouti allele. So because they don't carry the AVY allele itself, there's going to be no confounding effects on their phenotype from activity of the actual AVY. So what we found when we compared these offspring to these offspring is that the obese-born offspring were a little heavier, but otherwise they didn't really have any overt metabolic phenotypes. And at first we were a little disappointed at the lack, what we thought was a lack of a phenotype, but then we thought, well, maybe the increase in body weight is evidence of a latent phenotype, and maybe if we challenged the mice, an overt phenotype might appear. So what we did was we stratified the mice into two groups, and we challenged the groups with a high-fat Western-style diet or left them on control. And what we saw is that the obese-born offspring, at least the males, on the Western-style diet showed altered glucose metabolism. So in this glucose tolerance test, if we give the mice a bolus of glucose, the mice get rid of the glucose more slowly if they were obese-born compared to the lean-born cousins also on a Western diet. And similarly, if we give them a bolus of insulin, they show an altered response to the insulin. But on a normal chow diet, the groups are indistinguishable metabolically, even in late middle age. So looking at the phenotype a little more closely, we also saw that the obese-born offspring have defects in lipid metabolism. So for example, their liver triacylglycerides and diacylglycerides are higher, and this is exacerbated by the Western diet. They've also got higher serum leptin than their lean-born cousins, and again, this is increased by being on a Western diet. And the serum leptin correlates with the increased body mass in the animals, indicating that it's an increase in fat which is producing the leptin. But I guess most dramatically, the obese-born offspring also develop this really severe fatty liver phenotype on the Western diet, steatohepatitis. And this is in contrast to the lean-born animals, which even on the very high-fat Western diet have almost normal liver histopathology. So we considered that maybe the molecular lesion underlying these phenotypes might be epigenetic in nature. So we looked at the cytosine methylation across the genomes of these mice. And here we really exploited this latent phenotype because we didn't want to analyse mice that had florid disease just in case there were any epigenetic changes as a consequence of the disease. So we looked at the mice that didn't have the overt phenotype, the obese and lean-born animals on a control diet. And so when we profiled cytosine methylation at CPG islands across the genome, that is near gene promoters, what we saw was that the lean-born animals had almost 2,000 genes that were different when we corrected for multiple testing with a p-value of less than 1%, where there were differences between the two groups. So these differences were tended to be small. They tended to be less than 1.5-fold in magnitude. But I guess most interestingly, they also didn't seem to happen at metabolically associated genes. So in fact, the loci that are altered by maternal obesity are clustered in genes with developmental functions. And metabolic loci are really notable by their absence. We looked really hard to find metabolic genes associated with this phenotype, and we just couldn't find them. So it made us wonder what might actually be going on here, what might be mediating the phenotype that we see. And I suppose the thinking in this field of fetal programming has really been influenced a lot by the great David Barker and his studies in undernutrition, undernutrition experiments in children who were born of, say, mothers who've gone through famine. And there's this predictive adaptive response that's been hypothesised where a child with undernutrition in utero has been adapted to expect scarce resources, but they come out into a first world country, their expectations are not met, there are abundant resources, which all sort of go to the hips, as it were, leading to metabolic disease. But what we see is that with an overnutrition stressor, essentially the opposite stressor leads to a very similar phenotypic response. Now, the response to overnutrition is clearly maladaptive, so we speculate that maybe what's going on is that there is a generalised damage to the epigenome that happens in response to fetal programming, and this simply impairs responses to environmental stresses across the board. And of course, in modern day society, a very prominent environmental stressor is overnutrition, and so metabolic phenotypes may be the phenotypes that are the most obvious. So if these phenotypes are being epigenetically mediated, we know that epigenetic changes can be inherited from one generation to the next. So we asked whether these phenotypes are heritable. So we did our experiment where we looked at the offspring of obese diabetic mother or lean mother, and we took the F1 offspring, the males and females, and we mated them to their lean-born cousins so that we could follow any gender-specific patterns of inheritance. So is the phenotype heritable? Yes, it is. It seems to be, at least when passed through the mother. You can see that the F2 mice have a similarly blunted response to a glucose bolus. And remarkably, this seems to be maintained into F3 as well. This is a full 2 generations away from the mice that were exposed to the initial stressor. And so it seems that this phenotype has been inherited not just into the children of the exposed mice, but also into their grandchildren. Although I suppose there's also there's always the possibility that there's been a serial programming by the mothers themselves developing some sort of metabolic phenotype in pregnancy. Although the mothers were maintained on a control diet and shouldn't have had any overt disease, the pregnancy itself may have induced, say, insulin tolerance, for example. That's something that can be tested and that we're going to test. But that confounder can be ruled out, the confounder of the intrauterine environment itself perpetuating the phenotype, if you look at effects that come from the father. And so that's what we did. We looked at offspring who had obese diabetic fathers and asked if they also had a metabolic phenotype. And because I'm showing it to you, you'll know the answer is yes, they do. So the male offspring of obese diabetic fathers are heavier than their lean-born cousins, and they also exhibit altered glucose homeostasis as well. So here they're showing they've got glucose intolerance relative to their lean-born cousins, also on the Western diet, and also they've got a blunted response to insulin as well. And this effect is heritable. So F2 offspring of obese diabetic grandfathers also show the same effect on the Western diet, and it looks as though F3 male offspring do as well. And it just doesn't seem as though the phenotype has been ameliorated at all between generations. This looks just as dramatic as did the graph in F1, which I think is remarkable. So what does this mean? Essentially that mice who have an obese diabetic mother or father, or a grandmother or grandfather, or even a great-grandmother or a great-grandfather, seem to be predisposed. They've got a hypersensitivity to developing impaired glucose tolerance when they're challenged with a Western-style high-fat diet. And what is mediating this inheritance? It's not genetic. All these mice are genetically identical. And so we're interested in seeing whether it's an epigenetic effect mediating this phenotype, this heritable phenotype, by looking at cytosine methylation profiles across generations. And I guess if this finding translates to humans, what it really tells us is that the inheritance of the legacy of parental obesity and diabetes could be a significant contributor to the current cycle of obesity, where obesity seems to get passed down one generation to the next. So it remains only for me to thank everybody involved in the work. This has been a real collaborative effort in the epigenetics lab, and I really am just the mouthpiece for all the work that's been done, especially by Paul and Sally and Alistair and Cheryl, who started everything off. So thanks very much.